everyone, good day, especially to my Mel Joan Dizon. I am Ruby Jane Pao together with my partner Miss Mary Crest Taguan. This time, we will answer the 4 questions in Lesson 6. Without further ado, let's proceed and start the discussion. The difference between light-dependent and light-independent reactions. In light-dependent reactions, energy from sunlight is absorbed by chlorophyll, and that energy is converted into stored chemical energy. The chemical energy harvested during the light-independent reactions drives the assembly of sugar molecules from carbon dioxide. Based on the discussion, light-dependent reactions is the stage 1 in photosynthesis, which solar energy or photons is converted into chemical energy, which are the ATP and the NADPH. Whereas light-independent reactions is the stage 2 of photosynthesis, and it is also called the Calvin cycle. Uses the chemical energy produced from stage 1 to build G3P, the precursor to glucose and other molecules. Well, photons are absorbed and utilized in the first stage of photosynthesis if chlorophyll absorbs a photon of a particular energy. One of its electrons jump up to an excited state. These chlorophyll molecules sit inside of a photosystem with many other chlorophyll in its ground state. It's represented on the blue circles, and this excitation state represented at yellow-green circles. After absorbing a photon, the yellow circles can get passed around from molecule to molecule with one electron relaxing back to the ground state, emitting another photon that will excite an electron in another molecule. Eventually, a photon may strike the reaction center complex, representing the red circle, which also contains 2 molecules of chlorophyll. After a photon strikes the reaction center complex, the high-energy excited electron, rather than relaxing back down to the ground state, after which an enzyme supplies the missing electron to the oxidized chlorophyll via a water molecule to get it back to its normal state, back to the red in color, which is how oxygen molecules are produced.
The plants manufacture or synthesize the food they need to stay alive. Begins with these 2 processes called the photosynthesis. So the both parts of this complex takes place in the chloroplast. The product synthesized in stage 1 requires a lot of help from the sun, or known as the light-dependent reactions. The light reaction takes place fast in the grana of the chloroplast. The stacks of thylakoid, which contain the 2 photosystems in their membranes, and both the photosystem 1 and 2 harvest light energy from the sun. The chlorophyll called P680 rapidly emits an electron that is gained by the electron transport chain. This action takes place time after time, leaving the electrons holes behind. The P680 must replace all of the electrons that escape, so the water from the mesophyll gives an electron to chlorophyll. The water gets split or photolysed, which enzymatically provides the protons, electrons, and oxygen, and now other chemical actions begin. The electrons one by one refill the holes were left in chlorophyll. The protons quickly take place in the thylakoid lumen or space. The oxygen atoms, 2 at a time, form an oxygen gas and release into the air. The electron from the photosystem 2 move down the transport chain and lose energy, which is gained by cytochrome or B6F complex uses the energy to bump protons, which translocate from the stroma to the thylakoid space. This is called the chemiosmosis that powers the ATP synthesis. As the protons pass these enzymes, it spins around, and this releases the energy which bind the phosphate with the ADP. When the ADP and phosphate bind, ATP is formed and has a special job to do the Calvin cycle or the stage 2. The Calvin cycle uses those products plus the CO2 to make a sugar or saccharide from which the plant's food derived. It takes place last in the stroma of the chloroplast, the fluid-filled matrix that's contained outside the thylakoid. The cycle begins initially with the sugar called the RUBP, or the starting compound which contains the 5 carbon atoms in this chain. The RUBP must bind to carbon dioxide or the CO2 which plants takes from the air outside. The CO2 is a gaseous state and diffuses through the leaf stomatae, then moves to the stroma which it will be fixated into RUBP. The rubisco is an anabolic enzyme to help the CO2 and the RUBP bind, known as the carbon fixation. Phase 1, or the sugar formation. A 6-carbon compound now formed, but it's unstable and doesn't last long. It spins in half and generates the 2-PGA, or the phosphoglycerates. Phase 2, or known as the PGA reduction. The ATP molecules gives the phosphate to the PGA. The visphosphoglycerates are produced, which NADPH will reduce. The visphosphoglycerates now are changed and they lose phosphate from their carbon change. This reaction provides the PGAL and glyceraldehydrates 3-phosphate, or the 3GP. Several products also derive as the 3GP are synthesized. The ADP, and it also creates the NADPH plus and phosphates. All of this can be used when the light reaction takes place again. Both of the G3P contain 3 carbon atoms in their chains. Those carbons will be utilized to make the RUBP, or the more G3P must be obtained to build a 6-carbon glucose chain. The phase 3, or known as the generation of RUBP. 10 molecules of the 3GP are recycled enzymatically. They are rearranged and phosphorylated and RUBP is generated. Well, by 2, where we start from the G3P will be synthesized when the plant takes place in the carbon dioxide. So the photosynthesis part 1 and 2 help make the G3P, and when they do, the plant makes glucose their major fuel and other organic molecules. As I have mentioned from the previous discussion, during the light-dependent reaction, the sunlight plus water produce an oxygen, ATP, and the NADPH. While in the Calvin cycle, or the light-independent reaction, uses those products which are the ATP and NADPH plus the CO2, or the carbon dioxide, to produce a sugar. The rubisco captures CO2 and attaches into 5-carbon sugar, or called the ribose bisphosphate, or the RUBP, or in short, the ATP plus NADPH plus CO2 is equal to sugar.